I think we've got starting to have quite an audience filling in. So I'll make a start on your introduction. So thanks everyone for joining us today, this morning via Zoom to welcome Kingsley Dixon all the way from Western Australia. So just briefly, I'll just remind everyone that we have applications for roles on the committee for next year for BioSoc are open if you want to send in a manifesto. Otherwise, of course, see the newsletter and website as usual. But today I'm very excited to welcome Professor Kingsley Dixon, who is an expert, I would say, on Western Australian botany and landscape restoration, and he pioneered a lot of the work at the Kings Park and Botanic Gardens in Perth, and now I believe works primarily on sorts of landscape restoration work. So you don't want to hear from me, and over to you, Kingsley.
Well, thank you very much. And it's an enormous honour to be speaking to you all. And I thought I would tease you a little bit with the dying embers of Tuesday with the sunset. And I'm very privileged to live on the west coast of this extraordinary continent and quite close to the city. So it's a great joy to be here. I titled this talk quite provocatively, and you'll see why with one slide. And it's Darwin, of course, has been my fan. I'm one of those people that read The Origin of Species, couldn't understand it when I was really young because of the English, but then got to learn it a bit more. And Darwin means so much in so many parts of all of our lives. And the extraordinary thing about his thinking and his travels and his writings was that he was generally spot on, but he was wrong with just one thing. We're going to discover that during today's presentation. But I want to take you back just a little bit further. William Dampier, 1699, may be known to some of you, but one of my great joys as a botanist was visiting the extraordinary Sherrardian Herbarium, which you have at Oxford, which has this extraordinary plant specimen in it, which is Sturt's desert pea. It's had no less than 10 different names since it was first collected. Of course, William Dampier was a buccaneer. He was exploring the riches of what the Spanish referred to Australia as the El Dorado. Of course, it is an El Dorado, but not for gold and silver, but for iron ore, being one of the largest iron ore provinces on Earth. But William Dampier, being a buccaneer, wrote about his travels, collected plant specimens, and that's one of only two of the earliest collected specimens of Australia that's still extant. And it was really great going to see those and sensing William Dampier being in the room. Of course, William Dampier's writings were the germ for Jonathan Swift's Gulliver's Travels, because he wrote about this land far away. That's another story for another time. A little about me. Probably none of you have seen that $5 note, but it made a great impression on me on the 14th of February, 1966, when we dumped the British pounds, shillings and pence and took on decimalised currency. And of course, we are now fully metricised, and I would say quite a sensible society in that respect. But that $5 note really impressed me enormously because I saw this image of this man. It had banks. And I thought, well, I don't know what a banks is. And I had these pictures of all these various fruits and seeds and some flowers. Of course, when I asked my grandparents, who were gardeners, they said, oh, he was a great botanist. He was on Cook's expedition of discovery, 1770. And suddenly I realised my love and passion of plants as a young child was actually something you could do and be so good at it that you get your picture on a banknote. So I thought, well, that's rather good. But I was more enraptured with the pictures of various native plants on there. Of course, we dumped all of those colonial trappings, except for the Queen, on our new $5 note, but we still have some of our flowers, albeit very stylised in the bottom left, unrecognisable wattles, and apparently from my bird friends, the birds don't match any known species, but that's artistic licence. that's where I began, and my journey has been a really interesting one, because I've managed to travel the world as a botanist, working in botanic gardens, working in conservation science, and learning lots of things. And I just want to pose this question to you about what does a bluebell wood in England have in common with Australian bushland? Well, that's Australian bushland there. And that's an area where we work. Well, the thing about an English bluebell woodland is the English bluebell is not English. Your English rose is not English. The London plane is not from London. English elm is not even English. And many of the orchids are in fact borrowed. They're things that are blown over from the continent, as much as all of my colleagues at the Royal Botanic Gardens Kew would like to think they have a high level of genetic endemism. In fact, many of those are recent arrivals. That piece of bushland that I'm standing in has almost 99% of those species occurring just in my patch. And the abundance of that flora we're going to learn about in a moment. Now, it's not that botany is static in the western part of Australia, and it's an extraordinary continent of great diversity. But in Western Australia, we are in a renaissance of plant discovery through the eyes of the molecular geneticists. Take, for example, these two orchids. I found them 4 years ago, growing on a high remote plateau near my farm, about an hour south of the city of Perth. I thought they were just common relatives of a widespread species, but some sort of colour variant. What was unusual is that the white thing in the top left, this white fairy orchid, was always only flowering around that white flower, which is an ancestor of a very famous flower, the kangaroo paw. It's called Conostylus. Growing in a similar area, but always associated with this pink shrub, which is a myrtle, was this pink form. Now, it took my orchid colleagues, Mark Chase and Mike Fay and Martin Christenhuis from the Royal Botanic Gardens Kew, who were visiting me. I said, I want to take you to my isolated plateau, very difficult to get to show you these orchids. And we went up there, and they're in their tens of thousands, and there's no other orchids. And the close relative of this is bright yellow. And we walked, we collected some specimens, we pondered, we had a Darwinian moment, because they both turned to me and they said, really, Kingsley, these are two new species. And why is that? Well, not only do they have a unique pollinator that co-pollinates with the mimic flower, the cottontail and the Swan River myrtle that you see here, but unlike its supposed parental species, they have the most exquisite fragrance, which is part of the way they bring the unique pollinator. So that was described just 3 years ago in a special issue of Phytotaxa. Equally, we work in our tropical north of Australia, and working with Carlos Magdalene at the Royal Botanic Gardens Kew, we're revising a number of our sensational water lilies. But this thing that we discovered with Carlos 4 years ago in their little billabong, and only in the one billabong in the back, is actually the world's bluest water lily, with the scent of freesias and apricots. It is a stunner. It occurs only in that one billabong, and we're about to name it as Nymphaea christiniae, after the philanthropist who funded Carlos and myself to go on this expedition of discovery. So we're still finding new species. Now, this doesn't mean that the continent is in this rapid evolutionary hyperdrive. It's actually a continent that is old and extraordinarily stable. In fact, it has one of the oldest catalogues of life on Earth. This is an area I know very well. It's affectionately called the North Pole. It's in the far north of Western Australia. It's called the North Pole because of its contrasting conditions to anything polar. It's searing hot. It will hit, in a typical February day, temperatures of 45 to 48 degrees centigrade. Very sparse rainfall. It's a desert landscape. But in those rocks that you can see there, nearby, they found laminated structures, which when they sectioned, and you can see those in the middle section just here, they were the lamellae that you find in existing living stromatolites today. When they went to those nearby cherts and sectioned them, they managed to find evidence of filamentous cyanobacteria in those rock, in the preserved and fossilized, that were around 3.5 billion years old. still sitting in rocks, sitting on these landscapes. Of course, the rocks that you see in the distance are made of almost pure iron ore, up to 70% iron content. And of course, this is part of the hint. This is a very old, geologically very stable, with very little recent volcanism, and it's been 250 million years for the southwest since our last glaciation. So how did this all look? How did this all operate? This is Gondwana. The years, millions of years are slipping by on the bottom left. You can see that at about the 150 million year mark, which is when angiosperms came into being, we were still connected, Australia to Gondwana. By 65 million years, Australia had well and truly disconnected. There was circumcontinental flow of oceans, and Australia did something the other continents didn't do. It drifted in splendid isolation, carrying with it a capsule of genetic material of the ancestral stock of all the flowering plants. That's the beginning of the story. I'm now going to share with you a little part of that story, because what we've now discovered is that things don't move very fast when you've got an old and geologically stable continent such as Australia. This is a place I know very well. It's called the Kennedy Ranges. It's about a day's drive north of the city where I work. And in those sandstone beds, a few years back, they pulled out a fossil, and it's a remarkable fossil. That's what it looks like. And today, as a treat, I've managed to retrieve from our museum that very fossil. So you can see the actual thing in front of you. Now, that'll get more significant in just a moment, because that's the seed capsule of a genus called Banksia, named after Joseph Banks. The little valves that you see sticking out are where the seeds are protected, and it's a remarkable structure. But what's even more remarkable is if we compare that to the present day Banksias, they're almost indistinguishable. I've got my example of a present day Banksia right in front of you here, which I've just collected from a nearby tree growing in someone's garden. The extraordinary thing, and I put that up the top, is when you get the engineering near perfect, you don't need to change. And so what we've done in this continent is accumulate species, because that little fossil on the left, called Banksia archaeocarpa, means old fruit, is extinct. It was growing in rainforest, and it was growing in the Kennedy Ranges before they were deposited as sandstone around 50 million years ago. Very few fossils in the past, except for conifer fossils, carry that sort of level of preservation of genetic materials. So we've seen very deep time, moderate deep time at 50 million years. What about more contemporary time? Colleagues of mine found an interesting meteoric crater north of Perth. This is a very large crater. It's a kilometre or so deep. It's done in a flat landscape. It's filled with essentially dust deposits. They put cores down, and this is part of the 67 to 90 metre segment you see in the top right. You can see these laminations that they got out. This is rocky material. All the little black bits, that's microcarbon. That's charcoal that's been deposited in the white bits of the sand in between. What was intriguing when I started working on this with my colleague John Dodson was, and we were looking at the 2.5 to 2.9 million years mark, and I don't expect you to see all these names along the axis, but they're all sorts of genera and families of well-known Australian plant families. And this was all really, really interesting, not for one thing being that where you saw the peaks in some of the pollen that we got from that core, you saw a decline in others. And these were in fact mini climate shifts that were happening. But look at how many times they happen. So it's been a continent of constant climatic revolution. And climate shift is not, doesn't create extinctions, it creates diversification. What we found even more exciting when we go to the spot near that crater, which looks like that, we actually still are able to find exactly the same species from some of those pollen that we had at around 3 million years ago. living happily on these landscapes. They are really extraordinary indicators of the stability that we're dealing with in this continent. So this is me stuck down in the southwest corner of Western Australia. We're one of 36 biodiversity hotspots globally. I share my life with a range of fantastic endemic families, heaps of endemic genera. My colleagues from Kew and Missouri Botanic Gardens and New York Botanic Gardens, when they come, get very bored when I say, you know, this is endemic. I said, everything's endemic in Western Australia. You don't need to say that anymore. But many of these plants are things that are extraordinary. The bottom right is a thing called Ecological monostachya. If any of the audience know this plant, then you win the prize. You get to visit one day. Why is that important? That's been shown now through molecular evidence to be the oldest ancestor of something you eat every day, which are the cereals. So this is what original cereals look like, and we have this ancestor. It has 2 genera with just 1 species in each genus, its and its own family. But let's look across to this one, the Dasypoganaceae on this side. This is what it looks like. The panel I'm showing in the bottom is from 1836. It's called the Ensign Dale panel. And it shows these sittings at a place called King George's Sound, where Darwin actually came in 1836. And this is a really interesting plant. We thought it was just a member of Australia's grass tree group of species, a genus called Xanthorrhoea. We gave it its own genus name, Kingia. Again, the molecular eyesight that we got on this species has been a revelation. We were not looking at a species that is a contemporary species, this landscape, but it dates back into its own order. And so we're the only place with its own order of flowering plants. And it dates back at about 120 million years, about 15 to 20 million years after the dawn of flowering plants. Now, for all of you who are very interested in the enigma of long distance dispersal, this has been sitting on our landscapes, probably slightly changing over those periods of time. Why did it never exit and do long distance dispersal? That is one of the abominable mysteries that we deal with today. Now, sorry, I'm going to share with you now a short Minute Earth video that we did with colleagues, which encapsulates what has driven that diversification. And then I'm going to explain to you just some of the processes that have driven that diversification. So this is a very dense video, but I encourage you, we'll load this presentation and you can look at it at your leisure. So it's describing why in places like Western Australia, with some of the most impoverished and barren soils, as Darwin described them, early attempts at colonisation said they're barren soils, but in fact, that's the very reason we have the diversity in these landscapes.
A soccer field-sized patch of forest in frigid Alaska has about 40 different species of trees, shrubs, and herbs, compared with about 70 in temperate England and 300 in the Amazonian rainforest. These biodiversity differences hold true for entire countries, too. England has 1,500 plant species, while tropical Guyana has 6 times that many. This superdiversity of tropical rainforests is only equaled in one other type of ecosystem on Earth: scrubby, fire-prone shrublands that grow in Western Australia and Southern Africa. These shrublands may not look as majestic as tropical rainforests, but in a given area, they're home to similarly stupendous numbers of species, which doesn't necessarily mean that the rainforests and shrublands are easy places for plants to live. In fact, both ecosystems owe their enormous diversity in part to the fact that their soils have critically low supplies of nitrogen and especially phosphorus, nutrients plants need in order to grow. The plant world's leading biodiversity hotspots are quite literally dirt poor. Logically, it seems like richer soils should support more species, but in nature, as in human society, plenty of resources doesn't necessarily translate into everyone gets plenty. In meadows, forests, and wetlands around the world, we consistently find more or bigger plants but fewer species where soil nutrients are highest. The fastest spreading species soak up most of the extra nutrients, which lets them keep growing super fast, which lets their roots suck up so much water and their leaves snatch up so much sunlight that other slower species actually get less of those resources than otherwise. So in rich soils, slower species die out, while the fast growers win big. On the other hand, poor soils don't provide enough nutrient capital for fast-growing plants to build their massive infrastructures and take all the resources. So poor soils inhibit the greedy and allow everyone else to scrabble by. We see this pattern in human society too. In any given industry, there's a far greater number of mostly small businesses in poor countries, while fewer bigger companies dominate in rich countries. But crummy soil isn't the only thing that helps super high diversity blossom. For example, beaches, mountaintops, and other places frequently ravaged by harsh weather or catastrophic events have poor soils and few plant species. The other major prerequisite for hyperdiversity is time. On most of the planet, glaciers regularly bulldoze away ecosystems and grind up mineral-rich rock, creating new soil perfect for growth, but not diversity. However, our high-diversity rainforests and shrublands have spent millions of years beyond the reach of the ice sheets, leaving their residents plenty of undisturbed time to evolve a wide variety of ingenious strategies for surviving nutrient poverty. Strategies that have allowed for the development of tall, diverse rainforests in wet, poor soils and scrubby, diverse shrublands in dry, poor soils. The human landscape also seems to follow a similar pattern, with the highest cultural and linguistic diversity as well as the greatest number of businesses in climatically stable places where humans have been the longest and where economic resources are scarce. So in some ways, the poorest places on Earth are actually also the richest.
So I think that gives you in a very dense couple of minutes, essentially what has driven the diversification that we have in the southwest of Western Australia. We get that in Southeast Australia, we get it in some of our rainforest systems, but essentially in Southwest Western Australia with about 8,600 native plant species, about 1,500 in all of the British Isles, it certainly has been a major driver. So, Darwin did actually visit Western Australia. It was 1836. He was at the tail end of his extraordinary journey of discovery. This is an image of the second exit of the Beagle without Darwin, which was a commissioned work by a colleague of mine. And that's the Beagle leaving King George's Sound. But Darwin was on the ship. He did small forays into the early settlement of about 50 houses. He was unimpressed with the quality of the settlers, unimpressed with the quality of the buildings. It was high summer, there were flies. He had suffered great seasickness and other maladies on the journey. He was homesick. He wrote prolifically to his wife, stating how he was going to visit her so quickly on arrival. So Western Australia to him was completely unimpressive. What's interesting is, had he bothered to walk just over a few hills, he would have come across some of the world's most unique mammal-pollinated plants. He would have seen 14 to 17% of the flora in the region pollinated by birds. The orange flower, which would have been in great bloom then, the world's largest mistletoe. It's a giant tree. It's considered our most spectacular tree, Noitsia floribunda, an endemic monotypic genus all by itself, and a hemiparasite. And he would have found an extraordinary diversity of flora. Instead, he collected some rocks, some shells, some mussels. He wrote about the geology, wrote about the poor soils, and then made his most famous statement as he departed, that he, in fact, to add to that, he never wanted to return to our shores. Now, Darwin did get it wrong on that occasion. I just wish one of us had been there to pick him up, take him in the four-wheel drive and show him some of that country, because it could have impressed him. Now, what is interesting, though, is I've been rereading some of the analyses of Darwin's diaries from that time, and he actually did put jottings in an essay written in 1844, where he spoke about the fact that Western Australia, though on the same continent as Eastern Australia, separated by a vast desert, but with highly impoverished soils, that this vastness and separation and his words were, had seemed to have created the opportunity for a Galapagos in Western Australia. So he made those notes. So we like to think, as he departed Australia and went up to Cocos, where he then studied corals and the ways corals grow and wrote his definitive work on coral growth, that just possibly Western Australia, hidden away in his notes, had started to ignite his whole interest in the origin of species. So this is the sort of stuff he would have found. More bird pollination than any other flora on Earth. Real arms race between floral development and getting their pollinators. Equally, there are more plants that exploit animals and animals that exploit plants. This is the most extraordinary animal. It's the honey possum. A colleague of mine has done all the energetics of this, and on a human equivalent, this animal consumes daily nectar equivalent to us drinking 290 cans of full sugar strength Coke. Why do they need that? They have hyper high metabolism, so high, being in the Dasyuras, that the male immediately dies after consummating the female. So they live barely 11 months. And so at this time of the year, the females are running around with their little pouch young, and they're the only males. All the actual males have long since departed. Very clever mechanism for surviving stringencies. I was also privileged to be the first person to record reptile pollination. Although this is well known in South Africa, this is the first skink pollination of Banksia species that we found. Now, had he also bothered to walk into the scrublands, these things that look so uninteresting, he would have found an extraordinary diversity in very small areas. The girl that you see in the central image is a Bavarian colleague. She was doing her PhD, and she challenged me to the fact that she said, this looks interesting, but it's not as rich as our Bavarian grasslands. She said, they are really wonderfully diverse. So what I did was I challenged her. I said, I will sit down in the middle of this scrubland, and with just leaning, we'll work out how many flowering plant species I can collect. We collected just on 80 species. With her Bavarian tail between her legs, she was suitably humbled by the flora and realised that indeed it might look all the same, but it's very diverse. And there's some of the plants that were within arm's reach. Equally, further inland, we have extraordinary plants, the world's only fully underground orchids. First discovered in 1829, the newest species just recently described 2 years ago that you can see up there that I named, Rhizanthella johnstonii. This is a plant that has decided to devoid itself of roots, leaves, and has no chlorophyll. It's fully dependent on the woody shrub that you see there. And what we've managed to do is to piece together the whole life cycle of that plant. The little red arrow shows the seeds with a special hair going out to find the blue fungus that's been stained blue in that image. And only with the right fungus connected to the right root system of the right species do we get a thing called epiparasitism, where this orchid is able to take the carbon and the nutrients from the orchid. And we've now done the tracing and isotopic studies to prove this, to get that relationship. Equally, being fully underground, they're hard to find. It's pollinated predictably by termites. It creates capsules that are underground. They're fleshy, highly fragrant. Long since extinct are probably the native fossorial marsupials that would have dug these up, eaten them, and transmitted the seed. And we've done controlled feeding experiments with those species to show they can pass viable seed through those animals. But to indulge you for just a moment in my most passionate of plant families, the orchids, and David Attenborough being a close friend, we worked together on the private life of plants. He wrote the foreword to a book on orchid conservation we recently published, and he very elegantly said, orchids surely are the most glamorous of plants. For me, they have been one of those things as a child walking through our bush where we have nearly 420 species in the southwest, a place of great discovery. But it's the pollination that's been so intriguing for us. And even the most uninteresting of orchids, such as this winter flowering greenhood, this is a brown species, has proven to be fascinating. And I want to share with you this little video footage of a future PhD student of mine who came to me 6 years ago to say, I want to do a little winter project. I'm here on holidays from Italy. She was an orchid fanatic, Daniela Scacciabarozzi. We published a lot of stuff together. She's got a PhD. And I said, well, let's work out the pollinator of this. People think that fungus gnat, that the female looks for nest sites to lay eggs. So she tested that, and then she came back with this remarkable footage. Have a close look. It looks like that female's trying to probe to lay eggs, but wait and you'll see the lip, which is touch sensitive to the weight of a fungus gnat, being flicked inside the flower. If you look closer, you'll see this insect being maneuvered by one-way hairs into the flower, and in about 3 to 4 minutes, it will exit with the dusty pollen. Now, what was interesting with Daniela, I said, wow, there you go. So somehow it's gone inside and laid the eggs. And she said, Professor, there's a problem. They're all males. Indeed, what we've discovered, this orchid produces a sexual pheromone that's irresistible to nothing more diminutive than a fungus gnat. And just 3 years ago, in amber fossils from South America, what did they discover? Orchid pollen on a fungus gnat from 40 to 55 million years ago. Fascinating. Things have been doing these things for a long time. That no longer occurs in South America, but we are the last vessel carrying those great evolutionary experiments. Now, in terms of pollination, it's hard not to, of course, speak about probably our most beguiling of pollination systems, which is the West Australian hammer orchids that you see here. Sorry. The hammer orchids are an extraordinary group of plants that attract these male-finnered wasps. They no longer use colour, they use pure sex pheromones to attract the female, or the male, sorry. And so good is the release of the pheromones that you essentially get an extraordinary and somewhat R-rated event. Notice the little tags. They are our mark and recapture for looking at pollen travel and traversing distances, very localized in these groups. What we've discovered is that these orchids and a whole range of other Australian orchids, and in the southwest of Western Australia, we have more sexual pollinated orchids than anywhere else. Of course, Darwin talked about the sexual pollination in your most wonderful offers. But what we've discovered here is that unique molecules, pyrazines, and they're the special glands that sit on that hammer, are produced. But what's intriguing is for each of the remaining 9 species, there's one species already extinct, they each produce their own particular chemical signature. So you can grow all of these orchids together and they will never hybridize because the wasp stays faithful. So faithful is this system and so refined that what we've discovered is the wasps are better orchid taxonomists than the best orchid taxonomists. Here's a recent study that we've just completed. This is Drachea elastica. It's a very rare species. Nothing very surprising. It's a good species in its own right. It occurs in both a northern population that you can see there. It occurs also in a southern population with a big gap in between. And we got suspicious. But when we look at the two, they're indistinguishable morphologically. We did the genetic work, genetically identical. They're essentially fingers on the same hand. When we went to look for the wasp, the northern one, we got classic Zaspilothinus gilesii, very classic. When we took the northern flower and presented it to the southern population, Zaspilothinus gilesii was still attracted. But when we took the southern flower and took that to the northern population, we didn't. But what we did was we attracted a completely undescribed new species of Zaspilothinus. Morphologically identical, different semiochemicals pulling different pollinators. Here was the first case of cryptic speciation occurring right before our very eyes. Now, as you will learn as you become budding publishers, it's sometimes hard to get these sorts of controversial cryptic species theories published. So we've had to delete the term cryptic speciation because it was considered unacceptable, because they said unless you can find morphological evidence, it's not a cryptic species, it's just the same species. I think, and hopefully I've convinced you, that pulling different pollinators where we will not get into breeding is certainly a big step towards strong speciation. So to change tack a little bit and to move a bit back to Darwin, I just want to show you and remind you that Australia, and this is a MODIS image that we downloaded that shows the greening of the continent during winter and the drying during summer. Notice the desert regions never, ever connect. That has meant the Southwest has remained in splendid, as Darwin put it, Galapagos-like isolation. Now, one of the things I did in my honours year, which is the first year before starting a PhD, is I worked on sundews. We've got more sundew species in the southwest of Western Australia than anywhere else. In fact, we have 90% of the world's sundews. I think Britain, you have 1 or 2 species. Anyway, it's useful, but not as much as what we have. That's the distribution. But when you think that little tiny green corner in the southwest of Australia has 90% of the sundews, you start to get an idea. Again, I remind Mr. Darwin, had he bothered to walk across those landscapes, he would have found the thing that he found most interesting. And I take this quote from his book on carnivorous plants, that he cares more for Drosera than the origin of all the species of the world. So he loved them. And he would have seen the world's only pygmy sundews. He would have seen the dinner plate sundew that essentially munches on, it's a size of a bread and butter plate, munches on unwary insects. He would have seen these spider nets that essentially act like drift nets. He would have also seen things such as this, the Albany pitcher plant, a plant that is an endemic monotypic family, totally unrelated to the tropical pitcher plants. He would have found inside those pitchers that they are identical morphologically to the structures of the tropical pitcher plants, but what we now know from the molecular evidence is they're totally unrelated. But if we look at that vomit that's inside the pitchers of the Albany pitcher plant, that's a typical regurgitated meal. It's a bit hard to see, but the little circle that I've put there, they're protozoans that have learnt to adapt to the peptic enzymes that are in those juices. But what's more interesting is it's now been discovered that there's a flightless ant mimicking fly. That's a fly that mimics an ant that lives, that is able to lay its larvae in the fluid, and that this, called Bodicis ambulans, lives around those pitchers because it then feeds on the iridiomyrmex that fall into those pitchers. Now in 2019, I had the great joy of visiting this place. Of course, every biologist needs to go there. No prizes for guessing. This is Down House, wonderful place to visit. I was taken on one of the tours. Fantastic, wonderful guides, just hallowed ground my first visit. I dwelled in, of course, the greenhouse that they refurbished, where Darwin did some of his experiments, which I knew as a young honours student, where he fed bits of egg white and rump steak and all sorts of things to his sundews, and postulated that in fact it seems as though they can dissolve animal protein and potentially it may be a nitrogen nutrition. What I did in my honours was what Darwin could never do. We made labelled Drosophila flies with the heavy isotope of nitrogen, so that their protein had the heavy isotope of nitrogen, and we were able to trace the nitrogen into the sundews and prove that Darwin was in fact extremely correct. And what you see there is a formal presentation from the curator of Down House to the friends of Down House of a copy of my 1976 publication, where we announced that we had finally proven what Darwin had suggested, and he was exactly correct. Now, to conclude with, is I think it's important we live in difficult and challenging times for the environment of the planet. Darwin, again, was so extraordinarily prescient in what he anticipated. Sure, in the statement that you see there, the kangaroo and the emu have not become scarce. They've adapted to our agrarian systems of agriculture, and in fact are in plague proportions over much of the continent. What Darwin didn't realise is that we were about to put 11 mammals into extinction, the highest per capita mammal extinction on Earth. We have something like 60 plant species extinct, and I'm about to share with you some of why that is so. This is Warwickshire, 400 to 1960, a map of remnant woodlands. What happened in England was essentially changes to landscape at a snail's pace. Species could move around, migrate, colonise and do things. What we did, this is the wheat belt of Western Australia, hyper-diverse, 1920 to 1984. A simple 60-year period, we did the same. There was no way for our animals or plants to escape. Essentially, what you see in that southwest corner should have been all dark green. It's light green because we've cleared almost 98% of much of that area. If I put England over that, you can see the scale in 60 years of clearing was colossal. If I then show you this little map, just in that southwest corner, we have 109 species that we now know that are critically endangered and likely to go extinct in the next 5 to 10 years. What is daunting for me as a conservation biologist is these are the landscapes that I am left with. We know across that landscape that you see, we have 2,000 species where we just don't have the habitat. And hence my life has become as a restoration ecologist. Banksia brownii we now keep alive because of disease through the injection of phosphite into the stem of this amazing plant. The underground orchid has now reduced to just a handful of plants, potentially just one plant. Essentially, Rhizanthella gardneri, I spent my early botanical career hunting for it. That's an image from me from the 1980s, looking and finding the plant. 1982, halcyon days, 180. 2019 reduced to just one plant. 2021, we're not certain. That is an image, hopefully not of the last plant, and this May we go exploring again. But of course, it's not just the plants, but it's the animals that also face a very uncertain future. I sit on a federal committee. It was 10 years ago that I commented on this, one of our smallest of our microbats, the pipistrelle Christmas Island pipistrelle bat, that it was in precipitous decline. It's a federal committee that provides federal listing for threatened and endangered species. It's an extraordinary little species. I'm about to share with you probably one of the most harrowing moments in my career when Zoos Victoria moved in to try and save this last bat. What we have done on our landscapes, Christmas Island is part of Western Australia, so it's one of our protectorates, is we've mismanaged. And we continue to mismanage. Take, for example, this is an image from the front of my house. The birds that you see there and can hear are Carnaby's cockatoo. We have driven them virtually to extinction. This is in a loop, by the clearing of their habitat on a scale unparalleled. If you look at that image to your left, that was the Banksia woodlands. They have developed their mandibles specifically to use Banksia fruits and very few other species. And we've reduced that now to the point where it's a threatened ecological community. We're now working on a program to rapidly try to restore vast areas of landscapes in Western Australia to overcome what is the impending extinction of this, one of our largest of our parrot species. Once, as a child, the skies would go black with these as they migrated. Today, we're down to just 7,000 remaining individuals and limited forage. The challenges for us going forward are large. Originally, when I started restoration ecology, I had colleagues in Europe who said, it's easy. This is in Regensburg. You leave a meadow, 1975 to 82, the meadow naturally migrates to a shrubland. By 2006, it's a woodland. It's easy. You just leave it with time. Tragically, in old, stable landscapes, you don't need to move around much. This is an area that you can see here in the central wheat belt, the most devastated part of the Australian continent, cleared for cereal farming, as you've seen, cleared in World War II. He never planted a crop, he went to war and never returned. It's called Hartley's Reserve. We've been monitoring that now for 40 years and nothing migrates. We face large challenges with the 8,600 plant species and the many animal species to bring them back. Now, this doesn't mean that the task is beyond us. It means it's all hands to the pump, and we're seeing extraordinary opportunities. And it's the community that are driving it. This is Gondwana Link. Google it, look at it. It's the world's largest attempt to reconnect fragmented landscapes in the hyperdiverse southwest of Western Australia. And I'm honoured to be part of the team that's been working for the last 20 years and will continue to work to rebuild landscapes to protect and extinction-proof the southwest from the catastrophes that we've wrought. It doesn't mean that we're just doing this lightly. We're bringing extraordinary technology. Currently, we can restore on the left using native seed a matter of 100 hectares a day. We've now developed technology to put it through nothing as big as canola seeders to repatriate farmlands where we can put back 1,000 hectares a day to native species using pelleting technology. It's not just simple technology. We're the first group to develop what we call super seeds and smart seeds. These seeds are embalmed in biologicals to help rebuild the biological diversity, dormancy breakers, stress agents, in this case salicylic acid, many of you will know as aspirin, and carotenoid, which is another story of my life, which is the magic germinating promoter that we found and discovered in smoke. So we're working hard as restoration ecologists to repair and rebuild those damaged landscapes. Equally, we're working with my most cherished of plants, the orchids, and we're using things such as mycorrhizas to push back the extinction. And I'm pleased to announce yesterday the EU are co-funding with the Royal Botanic Gardens Kew a program to understand how climate will affect the mycorrhiza that you'll see here in David's footage from the private life of plants.
They found that many orchids have their own special fungal partner. They devised methods of isolating that fungus and then culturing it with the orchid seed. Under the right conditions, the two strike up their partnership immediately. The fungus extracts nutriment from the culture medium in a way that the orchid can't do for itself and supplies it to the young plant.
So we've been able to take exactly that technology, refine it, and we're now using it to rebuild many of our rare orchids. We've got a host of community volunteers across Australia that are helping to do this. We're pushing the technology to the very limits. And with this, we're seeing orchids rebuilt. We're also working with schools. We've developed a little kit where you can get your own little fungus, a little seed. You can sprinkle it on and grow your own seeds. But in this journey, we're not forgetting some of the most important and often forgotten components of Australia. It's our Indigenous people. This is something, probably my proudest achievement, which is the establishment of our very first Indigenous owned and operated native seed orchard on abandoned farmland where we're developing seed farming so we can generate the seed in time to ensure that we protect and enhance the bushlands that we need to restore. We need hundreds of tons of seed working with our Indigenous communities with resilient business models is certainly the next important part of my journey. Now, to conclude with, I really want to ask you a question. What does aluminium foil in your kitchen have to do with saving the world's greatest living laboratory? You're probably unaware. If you look at this image, this is, and you'll see the date stamp. This is Alcoa, the Aluminium Company of America, operating under a 1960s mining approval, who have blanche carte to clear the last remaining dry, temperate jarrah forest in the world. They clear 7 square kilometres and take away 12 metres on average. 11% of the world's aluminium, the aluminium foil that you use, contains aluminium undoubtedly sourced from our forest. What does this mean? This is a plot that I've done for tonight's present. Oh, and they're trees that have been removed, and there are up to 10 trucks a day move out of those forests. Carnaby's cockatoo, red-tailed cockatoos, and Baudin's cockatoo need the hollows that you can see in those logs. We hold grave concerns for the future. What does that cumulative footprint look like? We've drawn this up to show you the scale and to give you some idea of the magnitude of what's happening with no end in sight. That's London. So we are clearing areas that I believe now need to be reconsidered. I look to people in Europe, the EU and beyond to help remind our governments in Australia we have to rethink the way we operate. And I look to you people to say, how is my aluminium? How are my resources? How is the donut economy working for me? Am I paying the true cost for what I produce? And I'll leave you with this little phrase from my hero, and hopefully he's inspired you as well. There is grandeur in this view of life, and it's our task now to keep this life for future generations. Thank you very much.
Thank you so much for your talk. That was really inspiring and very interesting as a something to hear about from someone who's worked firsthand on this work. So I think we have some time for questions, if that's okay.
Yep.
So the first thing in the chat from Eloise, if you want to unmute yourself and give it out, if you want to. Let's see if. Okay, so Eloise has asked about how you deal with fires in the Gondwana Link program.
Ah, the thorny question of fires. Fire that happens in our landscapes has been very Europeanized, Eloise. We arrived with an English mentality. Remember, Elizabethan England burnt Ireland, it burnt. The moors are an artifact. We just like burning stuff. And fires are a natural but infrequent occurrence. And you might think fires occur everywhere all the time in Australia, but you're talking on a continental scale. So we have dates now coming out of our ecosystems where we know fire frequencies can be up to once in 80 years, now subjected to European style fire on once every 6-year rotations. That's not appropriate. So today I've in fact been talking to my Indigenous colleagues. We are now pushing for traditional burning, it was essentially called barefoot burning, where they go into landscapes and they burn small parts of it to get regeneration, but leave the bulk. What happens if a fire does occur? If we don't have weeds, and remember we have around 1,800 weed species waiting for fire, that's their entree into establishing a foothold in our nutrient-impoverished soils. If we control the weeds, then we're able to get regeneration and get it very well. But fire, used strategically, through an Indigenous lens, will be beneficial. I'm just going to go to my battery, my charger, so bear with me for a minute.
And Eloise has also asked about whether you have any tips or recommended reading in orchid taxonomy or orchid care.
Orchid taxonomy, You're a brave lady, Eloise. It's probably,
Yes.
I've done a little bit in orchid taxonomy and suffered the wrath of the orchid taxonomic mafia, but I would strongly suggest that you make contact with Mike Fay at Royal Botanic Gardens Kew and say where you're from and that you're very interested. They do amazing stuff in orchid taxonomy. So Mike Fay, m.fay@q.org.
Thank you very much. No, I get my reduced to clear orchids from my local garden centre for a £5, which is quite quite nice. But it's a bit difficult when you have one plant which likes 16 degrees and another which likes 23. So
yeah.
Thank you very much. I really enjoyed your talk.
Thank you.
So we also have a question in the chat about your thoughts on the role of the Indigenous community in wildlife conservation.
The role of the Indigenous community has been completely blindsided. For over 200 years, we have ignored them, we have dispossessed them, we have considered their knowledge irrelevant and not of relevance to the contemporary economic models. How wrong we were. We are now on a new journey in this country, as I think globally, in respecting First Nations knowledge and giving them the respect they are due. We're about to embark on a major program, hopefully to soon be federally funded, called Healing Country, which is Western science being led by Indigenous leaders to guide us in our scientific thinking. And it's humbling, to say the least. What you find with Indigenous Australians, they are quietly spoken, incredibly polite, generally will not push themselves forward. But once you get the dialogue going, it's one of the most enriching experiences. I had the great pleasure to be the only white person at a Circle of Elders meeting in November last year here in Perth. I was intimidated and I said I shouldn't turn up, and they said, no, you understand us, we want you to come along. And for 2 days, the Circle of Elders spoke, and I learnt every minute of that time things about my country, the way respect for ecology should happen that I'd never really fully respected. So yeah, it's a fantastic new journey I think the world is now facing. And the COVID pause, as I call it, that we're having is reconnecting people with nature and showing a new respect for fellow humans. And let's hope it stays. Not COVID, but the pause.
So if anyone has questions, so there's a question about from Mary, if you want to read it out, or I can read it out.
Sorry, I was just typing it in so you knew what it would be about. First of all, thank you so much. It's really interesting. I was just wondering, when you're doing the habitat restoration in the Gondwana link, for example, how do you decide which seeds you use? Problems associated, given that, like you were saying, with sitting down and having 80 species all around you, when you have like a lot of local endemism, I'm assuming that means that you don't, there'll be some species that are only found in quite a small area. Are there problems with spreading just a handful of species over a massive area?
So Mary, spot on. In the past, old-fashioned ecological restoration, 1990 style was, putting back 20 species was putting the bush back. It wasn't. We failed. We have, in fact, simplified these landscapes. They no longer support the diversity of animal life, and they certainly don't support the complex ecological functions that you need in soil and hydrology and so on. And Murray, what we've been working on all of my career, we've got a large group that work on what's called the Seed Enablement Program. So we've got 8,500 species in the southwest. Not all of them are needed in restoration, but we need probably around 2,000 to 3,000 species to be seed ready to be put into programs. Any one single area will have a minimum of around 300 species need to go through that big automatic seeder that we saw. We're getting there. we currently can put around 130 species through. So we're confident we can ultimately get to those numbers. These are numbers that are death-defying for my colleagues who do prairie restoration or people who do chalk grassland restoration in Britain, for example. The number of species is quantums of magnitude less. But the challenge is there. We've got the technology, we're developing that, and we're about this winter, which is in about 5 months, to do the first experimental plantings using large-scale industrial seeders. The same equipment that industrially cleared our landscape, we're now going to use to rebuild it with hyperdiverse species. Where we get local endemism, that's celebrated by looking at particular parts of the landscape. So we rarely have an endemic sitting in a piece of flat landscape. It's generally on some part of that landscape. It might be a piece of laterite, it could be serpentine, it could be a slight depression. So you will create those niches, or you'll find remnants in the, and you'll use those as the focal sites.
Cool, that's amazing. Thank you very much.
So following on from that, I was wondering how you approach genetic diversity in these in your seed supply.
Yes. This will offend many geneticists, what I'm about to say. Provenance, the issue. And we've worked in genetic provenance, and when you look at the literature that's published, we generally publish the exceptions. We publish the things where you show that provenance is critical and fundamental. And there's a few textbook examples we've all learned, and I learned when I was doing textbooks, that it was so important to represent this genetic diversity only in that site and so. And sure, that holds true for lots and lots of species. But to slavishly say that your catchment for genetic material should be only 5 kilometres or 20 kilometres, what we've now found, and it's being published at an increasing rate, is often the catchments are hundreds of kilometres. They're not as limited as we thought. Of course, people will then say, what about climate adaptation? The great thing for us on our landscapes is the evidence that I showed you from the impact crater from 2.9 million years ago. These species have been sitting around on the landscape. Sure, they've probably got a little thinner, probably slightly dwarfing as it's got dry, but they didn't go extinct. And so we're comfortable in the main that the predictions that we had for the Southwest of wholesale climate extinguishment of whole plant groups based on modelling didn't take into account the inbuilt thermal plasticity that we have in our species. So I think we're in a very confident space. Now that's very different for you people in the UK. You have very thermally bound species that need to move up mountains or move down or go somewhere else. So very different situation, but certainly in these old environments, it appears that things may be a lot more stable.
Really interesting, actually, to think about that sort of difference. And so we have a question by Giovanni in the chat. Would you like to ask your question about pollinators?
Yeah, sure. Thank you so much for your talk. I was wondering if there is any reason you can think of for the diversity of vertebrate pollinators in Western Australia, whereas insects, for example, are dominant elsewhere.
Oh, it's only 14 to 17% of our flora use vertebrates, the rest use insects. And wind pollination is quite rare for us. So for example, we only have one wind pollinated tree species. All of Europe, nearly all your native trees are all wind pollinated, the oaks and the elms and the birch and so on. So insects are a really big deal for us. And often you will find, although there is vertebrate pollination, mostly birds, the honey eaters as they're referred to, you'll find often, and this is a paper just published a few weeks ago by a colleague, Steve Hopper, and one of his PhD students, showing that you could in fact get surrogate pollination by other insects in typically bird pollinated plants. Sure, often lower seed set, but you will still get it. So even if you don't get vertebrates, they seem to be able to bet hedge with some seed production via insect systems. What we have found, though, is the vertebrate system is a very clever system, because birds are territorial, particularly the honeyeaters, that we can see microgenetic diversification over hillsides. So on one side of a hillside with a kangaroo paw species, which is predominantly bird pollinated, you'll find that the birds there are very territorial and they don't move pollen across the other side of the hill. And so you find slight genetic differences in the two subpopulations. Morphologically and in a restoration sense, they're indistinguishable.
Thank you very much.
Thank you.
So if anyone else has questions, feel free to send in the chat and I'm happy to read them out, or you can unmute yourself and ask Kingsley directly. But I was wondering where would you recommend people look if they're particularly interested in your work and this sort of your restoration projects? Do you have a particular papers or sources you recommend?
Gosh, I just updated my publication list today. It's getting fairly long. That's available, but it's yeah. You know, I guess what you're saying is there a book that synthesizes all of that? And, you know, my life in botany sort of thing. I think that happens when I'm dead. Someone else writes that. But we've got yeah. I've written a couple of books. One of my great interests is coastal restoration. We just published that last year. That's relevant only to Australian coastlines. So, and there's a few other books. In orchids, there's lots of stuff that we've written, particularly books that people can source. Just look at my name and they'll come popping out. And yeah, always an embarrassing question, I find.
So we have a question in the chat about agroforestry, and if you have any thoughts on agroforestry and habitat restoration.
Sure do, yes. We have to be very cautious. The push to put carbon into trees at the expense of biodiverse carbon will lead us down a tragic path. There's a number of key publications being done, not by my group, but by other groups, showing biodiverse carbon through biodiverse plantings and not monocultures or even polycultures of trees, where you've got understory, where you've got herbaceous layers, is the smart way to actually get the biodiversity bonus and more resilient carbon built up in the soil. We're just starting a big grant, a national grant, to map how that happens in various levels of remnant biodiverse areas to look at the carbon sequestration capability. But I urge great caution. Forestry is rebadging itself. Forestry has caused almost a third of the loss of plant cover on the planet. So that's a thing that came out in Nature, published 6 weeks ago. I don't have the reference with me. And forestry and forest management is responsible for the significant part of our plant loss, our plant biomass loss. Be careful how it's being repackaged to further cause problems on the planet. Sure, trees can be put in, but trees need to be there forever. If you chop them down and do biochar or all these other exploitative methods of burning biomass to create energy, we are just fueling carbon emissions. So as part of that, the UN is, of course, this year embarking on the Decade of Restoration. That was launched in January. I'm part of the UN group, and I've enjoyed very much keeping them sharply focused on the fact that it isn't just about tree planting. It's about the broader biodiversity reinstatement that we need to do across the planet. And I hope you will all be on that journey. It's not just the wood for the trees, it's the full complement of biodiversity.
Thanks very much. If anyone else has any questions, feel free to put them in the chat or unmute yourself. Someone has asked whether you'd be able to, would you whether you'd be able to send the slides out for people to have a look at
Yes.
in the future?
Yeah, so what I'll do is I'll Dropbox them to you.
Yeah, if you send them to me, then I can distribute them in our mailing list.
Fabulous. Yeah. They'll be in a PDF, if that's all right.
Yep, that's great.
Thank you.
So I'll send that around in our email list for people who would like to have another look at the slides.
Yeah.
And so there's a mailing list. If you're not on the mailing list, send the page a message and I can add you on Facebook or by email.
Thank you. And look, I guess, everyone's asking about a copy of the recording.
I'll sort of, are you happy for me to distribute a recording of the talk just on the mailing list to members?
Yeah.
Great.
Good.
So I will distribute that by the mailing list.
Yeah.
Great. So thanks very much for speaking to us today. It's really a very inspiring talk to have you presenting your work, which I think seems to be really important, and wish you the best of luck with all of the work that your group is doing in landscape restoration.
Thank you.
So thanks very much, Kingsley. If anyone would like to turn on their cameras and say goodbye as they leave, just to sort of...
Fantastic. Oh, lovely.
Thank you.
Thank you very much.
Thanks very much.
See you.